Assalamualaikum and salam sejahtera. I am Huwaida Muhammad Hanifah, S50685, supervised by Dr. Siti Sofo Ismail and co-supervised by Dr. Nurul Shahida Rizwan, would like to present my final year project proposal titled Diatom Diversity Associated with the Decomposition of Partially Submerged Cadaver Buried in Coastal Area. For background of study, many methods have been used to locate the clandestine grave, for example, cadaver dots and geophysical surveys involving soil analysis. Next, the properties of diatom species and its diversities can be used in determining the location of clandestine grave. The buried body will undergo 5 stages of decomposition process, which help in organizing the postmortem changes and any information related. Turning now is to my literature review. Decomposition is a process of breaking down organic matter into smaller constituents. Their characteristics will help in organizing postmortem changes, and the 5 stages of decomposition process depend on the environmental factors. So here is the 5 stages of decomposition process, which are initial decay, putrefaction, black putrefaction, butyric fermentation, and dry decay. Diatoms can be everywhere in environments such as marine and freshwater ecosystems. The diatoms are unicellular and eukaryotic microorganisms with length of 10 to 100 micrometer. The diatoms shall provide significant information that may aid in locating the clandestine grave or the postmortem interval. Decomposition process depends on intrinsic and extrinsic factors. Intrinsic factors include age and microbial activity, while for the extrinsic factors, which divided into 2 parts, biotic and abiotic. Abiotic include temperature, humidity, groundwater, soil type, and pH, while microbe is a biotic factor. The problem statements are the study of partially submerged cadaver buried in a coastal area is lack of research compared to those in terrestrial area and drowning water cases. The inconsistent variables may alter the decomposition process due to the season change, and this will lead to difficulties in estimating the postmortem interval. Significance of study is decomposition process and diatom diversity contributes useful information to locate the clandestine grave. Next is estimating the PMI, subsequently solving the criminal or murder cases. This study will lead to more comprehensive understanding on the decomposition process of partially submerged cadaver in water bodies of different salinities. My specific objectives of this research are to elucidate the decomposition rate of a partially submerged cadaver buried in a coastal area and to determine the diatom species associated with the decomposition of partially submerged cadaver buried in a coastal soil system. There are 3 steps in methodology. The first one is simulated burial experiment. It will be done by recording the mass of 20 gram abdominal fatty flesh of commercial pig first. The pot containing abdominal fatty flesh will be covered with the soil on top layer of fatty flesh and then will be partially submerged in a coastal area at certain depth. Each burial interval will be replicated along with the control soils without fatty flesh. Diatom sampling will be done by collecting the associated topsoil at each sampling points to extract the resided diatoms. Next, each 10 gram of topsoil collected will be put in centrifuge tube and added with 1% of formalin. The sample will be centrifuged at 2300 rpm in 15 minutes. Distilled water will be added to a depth of 0.5 cm before agitation process. Half of it will be removed and placed in a plastic bottle contains formalin. 1 ml of potassium permanganate will be added into remaining agitated solution and left for 24 hours. The concentrated hydrochloric acid will be added in the samples and placed in a water bath at 80 degrees Celsius for an hour. The tubes will be centrifuged again for 15 minutes at 2300 rpm, and then the supernatant will be siphoned off, replaced with distilled water. Repeated centrifugation is required to ensure the clean of diatom sample. To prepare the diatom slide, white layer or diatoms in centrifuge tube will be blown and then it will appear on top of the soil within the centrifuge tube. 1 ml of distilled water in the tube will be transferred into slide chamber. Then 1 drop of iodide will be dropped within the slide chamber and leave for 2 hours. Cover slip will cover on top and the excess solution will be removed with a tissue. The slide will be observed and the species will be identified. To analyze the diatoms, we will use both magnifying microscope and scanning electron microscopy. The expected results are the decomposition rate will be slow at the early stage, quick in intermediate stage, and be moderate again at the end of the experiment due to the complete decomposition of body tissues. The number of diatoms too will decline in the later stage of decomposition due to large amount of nutrient released from the cadaver. Enclosed is my reference and Gantt chart. Thank you for watching my presentation video.